dear friends, today we are going to continue next part of embryonic development, that's gastrulation. So let's continue. As gastrulation is the process of formation of gastrula from blastocyst. In gastrulation, the rate of cleavage or division is very slow, but there are two important events that takes place actively. The first event, that's a differentiation of blastomeres, and the second one is a morphogenetic movement. The first event, that's a differentiation of blastomeres. This process results in formation of the three germinal layer, that is ectoderm, mesoderm, endoderm from the cell of embryoblast. The morphogenetic development is the second event. These are different types of movement to reach their definite place in the embryo. As in the last phenomena, it's a blastolation. In blastolation, there is a two layer is there. The first one is a trophoblast cell and the second one is a innermost cell. As this trophoblast cell, it undergoes a division and there is a formation of the two layer of cell, that is the syncytotrophoblast and the second one is the cytotrophoblast. As this syncytotrophoblast, it gives out the processes. It gives out the processes which is extensively entered invade it into the endometrium region. Result is what? This blastocyst, it is completely burrow in the endometrium, and the embryo, as this embryo, it get implanted into the endometrium region. As inside to it, there is a presence of innermost cell. This innermost cell, it undergoes the differentiation of the two layer of the cell. First one, it's a hypotrophoblast and the second one is the First one is the epiblast and the second one is the hypoblast. This epiblast layer, which is towards the amnion, and the hypoblast cell, which is towards the yolk sac. As this embryo, it is connected to the stalk-like structure that is called as the connecting stalk. So what about the development of the extraembryonic membrane? On about 8th day after fertilization, cell of the free end. So this one is the free end and this one is the endometrial region, and this region it says as the embryo. As the cell of the free end of innermost cell, that is called as the hypoblast cell, means this hypoblast cell which is towards the free end. This hypoblast cell, it becomes flat-ended. So you can see here, this cell, it becomes flat-ended cell, and it is start dividing and grow downward towards the blastocele. This one is a blastocele. This hypoblast cell, it is divide and grow downward towards the blastocele. That's the cavity of the blastocyst. It grows within the blastocele, and there is a formation of a structure, and that structure, it is called as the yolk sac. So you can see here, as there is a presence of this cell, that is a hypoblast cell, it divides and it forms a cavity, that is, it forms a sac, and that sac, it is called as the yolk sac. The remaining cell of the innermost cell, which is in contact with the stalk, that is the cell of rubber, that is called as the epiblast cell. This epiblast cell, it is a function as a primary ectoderm. Both the layer form a flat bilaminar embryonal disc. So these two layers, it together forms a flat bilaminar embryonal disc. After the formation of endoderm, the second layer to be differentiated is the ectoderm layer. The cell of epiblast. So This is the cell of epiblast. The cell of epiblast, it is divide and redivide and move in such a way that it encloses this amnion cavity. It encloses this amnion cavity. The roof of amniotic cavity, it is lined by the amniotic cell and the floor. So this one is the floor of epiblast. The floor of this has the embryonal disc. Means here there is a presence of embryonal disc and here there is a presence of the amnion cell. Afterward, the amnion cell, it is divide and redivide and there is a formation of the amnion. This amnion is an extra embryonic membrane that surrounds and protects the embryo. As a result of all these changes, the bilaminar embryonic disc is positioned in between the yolk sac. So this is the embryonal disc and this embryonal disc, it is present between the yolk sac and the amnion cavity. So afterward we see the process of gastrulation. So what's the process of gastrulation? This gastrulation occurs about 15 days after fertilization, in which the bilaminar embryonic disc, this bilaminar embryonic disc means which two layers? The first one is the hypoblast and the second one is the epiblast cell. It is transferred into the trilaminar embryonic disc, and this trilaminar embryonic disc means the ectoderm, mesoderm and the endoderm. This three embryonic disc, that is a trilaminar layer, is formed. This transformation, it is occurred by division and the redivision and the migration of the cell of the epiblast. So you can see here that is there is a presence of the two layers. That this two layer, that is the hypoblast and the epiblast. This hypoblast and the epiblast, it is formed from the innermost cell. That is the cell of innermost, it undergoes the differentiation and result is the formation of the epiblast and the hypoblast cell. This epiblast cell, this lower cell, it says as the epiblast cell. This epiblast cell, you can see this epiblast cell, it is migrated into midline to form the primitive streak. So this region, it says as the primitive streak. This primitive streak and a shallow groove. So you can see here there is a formation of the groove on the surface, that is called as the primitive groove. It called as the primitive groove. The streak, it is progressed from posterior to anterior end of embryonic. As this epiblast cell, it is migrate it into the midline and there is a formation of this region, that is says as a primitive streak. The primitive streak, it is elongate from caudal to cranial. Afterward that, some cell of epiblast, it is migrated into the primitive streak and some cell, it is enters it into the hypoblast. So this region, it says as the hypoblast region, and in this hypoblast region, some of the epiblast cell, it is entered and some of the epiblast cell which is remain present in the lower side, which is towards the amnion cavity, Some cell of the epiblast, it remain as it is and the some cell, it is enters it into the hypoblast cell and the some cell which is present it into the primitive cell. What happened after the transfer? These the epiblast it into the hypoblast. Result is what? There is a formation of this endoderm. There is a formation of this layer, that is says as the endoderm, and the remaining cell of the epiblast cell which is function as the ectoderm. Afterward that, majority of the epiblast cell which is occupy the middle space. Okay, majority of the cell, it's occupy the middle space and that space which is present between the epiblast and the hypoblast. That's a differentiation forms the mesoderm. The most important phenomena is that is the notochord is a first mesodermal tissue which is formed from, that is the notochord which is formed from the mesodermal tissue. Afterward that, so you can see in this image there is a presence of the primitive streak. This primitive streak which is formed from the epiblast cell and which is formed towards the amnion cavity, and opposite it there is a presence of the yolk sac. So this one is the yolk sac. Afterward, what's this phenomena we already studied, that is the process of gastrulation. The gastrulation occurs about 15 days after fertilization, in which the bilaminar embryonic disc, that is this bilaminar layer, it's a hypoblast and the epiblast, is transferred it into the trilaminar embryonic disc. The transformation occurs by division, rearrangement and the migration of the epiblast cell. Means which of the cell it undergoes the migration, that is the epiblast cell. It's a very important phenomena here. And the result is what? There is a formation of the primitive streak. So you can see here, this is the primitive streak itself. There is a formation of the shallow groove on the surface. That's a primitive groove is formed. And this streak, it is progressed from anterior to posterior end of embryo. Afterward that, the anterior end of primitive groove communicate with the yolk sac. Means this groove, it is communicate which region? That this region it says as the yolk sac, the cell of epiblast cell. So this one is the cell of epiblast cell which is move inward below the primitive streak and detach from blastocyst. Some of the cell it is invaginate this place, the hypoblast, and result is what? As the sum of the cell of epiblast, it is entered it into the hypoblast region, and result is what? There is a formation of the endoderm region, and the remaining cell of the epiblast is result it into the formation of the ectoderm layer. From the site of the primitive streak, the third layer which is formed, that is the endoderm region. And after the formation of this ectoderm, mesoderm and the endoderm, for the process after the gastrulation, that is called as the organogenesis. So the next phenomena we see, that is a difference between the blastula and the gastrula. In the blastula we already studied there is a no movement of cell, but here in case of gastrula result from morphogenetic movement of cell. The blastula it is occurred, it's a formed from morula, and the gastrula it's a formed from blastula. The primary germ layer are not formed, but here in gastrula there is a formation of the three germ layer. This three germ layer, it's a ectoderm, mesoderm and endoderm. In the blastula there is a presence of cavity that is called as the blastocoel. It's a archenteron. It's not formed, but here there is a presence of cavity that is a archenteron. It's also called as a primitive gut. The blastocoel, it's a highly reduced in case of gastrula. The blastula, it's a generally hollow ball of a cell, but gastrula, it's a two-layered structure. Hope so you understood it. Thanks for watching my video. Stay tuned to my channel. Thank you.